So can you hear me all right there? Yes? Yes. So yeah, well, welcome to the first of 2 round tables tonight. This is an experiment. Many of us are experimentalists, so why not? But many of the questions that we're going to talk about will probably be repeated tonight. The program is in 2 parts. In the first part, each panelist will be invited to give a short statement on how they believe on the questions that are addressed, like can we understand biology from purely molecular perspective and what's the role of physics, and then everybody should have his own freedom to talk they want. And in the second part then, we will ask questions and we will see how the discussion follows, and then at the very end we probably will include the audience as well. So this is what we have to offer for you tonight, and we will see how this goes. But we are definitely as curious as you are. I will start with saying a few words about everybody, if you don't mind, and start with the gentleman over there on the right, Professor Jean-Pierre. His work on allostery from the 1960s, as well as the acetylcholine receptor, are milestones in science. I teach, and part of those things I teach in my classes, in my courses. Although he's by training a molecular biologist, I believe his knowledge and interest is extremely broad, very broad. And I even had to find out that he had a long time ago published with somebody that physicists know from solid state physics, namely with Charles Kittel, a quite theoretical paper on believe it or not, and in the last paragraph he writes about propagating conformational transitions in membranes. So an idea, of course, that I'm a big fan about. It was a quite theoretical paper, although, as I said, by training a molecular biologist. I imagine his work on synaptic epigenetic and global workspace that he's going to talk about tomorrow probably more is again from another perspective, looking at the entire brain, not on single molecules. So I think that his background is much more, or his expertise is less defined by a discipline, but much more about being a creative mind. So please join me and welcome Professor Jean-Pierre from Paris. So the next in line is Thomas Heimburg. He did his PhDs on phase transition and phase diagrams, but if I remember correctly, this were membranes then, there was metals, if I remember correctly, but it was...
No, I did my master's on theoretical solid state physics.
The master's.
And then phase diagrams, that was more in my PhD with membranes.
Right, okay. So he, nevertheless, what is it? No, okay. So he continued his work on thermodynamics. I learned a lot of thermodynamics from him during my time at the Max Planck in Göttingen. And he went on then after his habilitation and after his Heisenberg stipend to join the Niels Bohr Institute, where he developed together with Heimburg-Jackson the soliton model that we heard lots about today. He's working on a new theory of anesthesia, as you know, as well as on the role of thermodynamic fluctuations in ion transport, in many more things, centered, many of them, around application of thermodynamics in biological systems. So please give a round of applause to Thomas Hanke from the Universität. And last but not least, we have an astrophysicist here. So Wolfgang Hanke did his PhD, I believe, in astrophysics. No, in astrophysics. But I would today more say he's a biophysicist and electrophysiologist. He did his PhD with Hohenheim in Bochum on black liquid membranes, learned a lot even from the famous Hermine, and wrote a great book that I can highly recommend, and now I got one of the last copies. I'm very happy about that. He worked then on the propagation of waves in the brain, spreading depression, and eventually, at least partially, managed to come back to his roots in astrophysics and works on the effect of microgravity or low gravity or gravity-free conditions on living systems. So a warm welcome to Professor Hanke from the University of Hohenheim. So I would ask, maybe in the same order, for a brief statement on how do you think and how far are we going to be capable to understand or should we go on understanding living systems from molecules and molecular interactions entirely, or do we need more? Or is it enough to understand individual molecules to come back to life?
First of all, we should say that we heard about models, and we are always producing models and testing them, and these models originate from the human brain, and they can describe adequately elements of the external reality, of our internal reality, but they are always limited. So I think that nobody, perhaps some physicists do, but the biologists have not the pretension to propose models which give an exhaustive description of reality. At least I never had this ambition. And the only thing we want to do is to produce models which match the reality, can be tested, and are going to give outcomes which may help us to further understand the system we are working with. So I think a model has not only to be adequate to reality, but to be productive. And this is, I think, one very important aspect of it. Now, concerning the question you raised of the importance of molecular biology. First of all, I would like to say that we are today having a distinguished population of scientists that you have already classified yourself as physicists, as neurophysiologists, as astrophysicists, and as molecular biologists. So I'm questioning this kind of classification. I think it has been useful at some time to get money, to get recognition in the academic, to fit with some of the big prize sections. But otherwise, it has, to my opinion, been very unproductive for science. To be clear on that, I would like to say that we need to have experts in all fields, like physicists, like molecular biologists, and so on. But we should avoid to classify them into a rigid structure. And for instance, the students we have now are both outstanding physiologists, outstanding molecular biologists, and computational scientists. They have to be all these things together. So my view is that all these different disciplines should interact to produce original and new and productive models. Now, I'm answering your question after this introduction. I think the the knowledge, I would like to say, the knowledge of the molecular level, because as was said by Nobel, and I will mention it tomorrow, we have to think about living organisms as being organized at several nested levels of organization, from the atomic to the molecular to the so on and so forth. So I think the molecular knowledge has made so much progress during the past decades that it's, I would say, it's part of the knowledge of the living organism, from the bacteria up to the human brain. And I would like to insist on one aspect, is that to my opinion, it's a necessary kind of knowledge. One cannot omit it. And unfortunately, it's not always the case. I realize that here there is some kind, I would say, hesitation. But when you look at some computer scientists interested in the brain, they simply deliberately say, we are not interested. We are having signals, and these signals, we describe them as spike or whatever. We describe neurons as points, and so on and so forth. And then we make a model of the brain, and I just heard about the question, if you replace a dot by a cow, what is going to happen? So it means simply that we have to have this basic knowledge constantly present in mind, and I think at this stage, I don't want to say that this molecular knowledge is fully known, certainly not. There are still many aspects of the molecular biology to be understood, but we should try to reconstruct progressively higher level of organization. And I like in your approach, and I stop now, for instance of the Hodgkin-Huxley model, that for me there are some aspects of it which are completely insufficient, I mentioned them, but also there are some physical events which are associated with it, which may be extremely useful to understand, to go up into higher level of organization. Sorry for being so long.
No, all the time, it's fine. Thank you very much. Thomas, do you want to?
Okay, I believe that the discovery of the DNA and this transcription into proteins of the genes is a fundamental revolution in science. It's very important. But I'm a profound hater of pathways, biochemical pathways, because I find it's closer. I believe that the biochemical pathways, they miss important aspects of reality, in particular the thermodynamics of the systems. If you look at a picture of a pathway, you see all these individual reactions, binary reactions, two proteins linked by an arrow, and maybe calcium binds or ATP binds. But there is practically no mention of physical laws in there, except for maybe a binding constant. You have no pressure dependencies, temperature plays hardly any role in these processes. There's no mention of entropy in there. So what a typical physicist's approach would be to complex systems like that, to say that you need models that are consistent with the scale on which the phenomenon takes place. So if you have a single molecule, an enzyme for example, it's very appropriate to look at enzymes if you want to study an enzymatic reaction. But if you want to study something that takes place on the centimeter scale or something like that, the molecular view doesn't cover the most fundamental aspects. As an example, I typically take a wave of water. You have a lake and a surface wave of water, and you ask the question whether you can understand the wave on the water by studying single water molecules. And every physicist would immediately say, no, that's not possible, because the single water molecule doesn't even see the changes that happen on the surface, because on the scale of a single molecule, there's practically nothing going on. And the thermal fluctuations are much larger than the things that emerge on large scales. And that is then the keyword. That's emergent phenomena on large scales that are not parts of the behavior of the single molecules. They emerge on larger scales, and they provide the thermodynamic couplings, the pressure and temperature dependence and things like that. So to answer that question that Matthias asked in the beginning, I believe that the concept of having single molecules is very useful as long as you stay on the single molecular level. And of course, the concept of waves or everything microscopic will not help you very much to understand an enzyme. But vice versa, the understanding of single molecules will not help you to reconstruct phenomena that happen on large scales, and because you cannot In the case of the water wave, you cannot construct the water wave out of the behavior of a single water molecule. If you study that for hundreds of years, and you study the vibrations in the molecules and the H bonds in the molecules, you will not be able to reconstruct the macroscopic phenomenon of a wave, because that is practically independent of the nature of water. There are also waves on alcohol or on completely different molecules. The feature of a wave is not a feature of the single molecule in there. And therefore I think one should look at what kind of phenomena we treat before we make a decision of how we treat them. In the case of the nerve pulse that we discuss here very much, we have said that the nerve pulse is several millimeters or even centimeters long. That is a scale that is several orders of magnitude larger than a single protein, like an ion channel. The scale difference between an ion channel and the length of the nerve pulse is about the difference between this cup here and the complete continent of Europe. So what basically, when we try to explain a nerve pulse with single ion channels, we claim that we can explain the phenomena that occur on the scale of Europe by just considering cup-sized objects, and I don't believe this is reasonable.
Thank you very much. Wolfgang.
Okay. Basically, I would go a little bit the same direction insofar that I think we have a very fundamental knowledge at the molecular level, by sure, and I agree by sure with the statement that to derive from this knowledge on the molecular level information about, for example, function of the complete brain, that there is a lot of information which will be messed in between, and there are a few very general questions which I would like to point out shortly. The first thing is something which is related to a historical aspect. You see, up to the 60s or so, the so-called question of excitable media or non-equilibrium systems was not really a point of view. For example, in terms of the Belosa-Sabotinsky, even in the 50s and 60s, it was ignored that these things exist. But meanwhile, from thermodynamical point of view, but also on general sciences, we know that these so-called excitable media or complex systems exist, and they should have simple certain properties, and you can make certain statements. One of the statements basically is that, for example, most biological systems will fall under the definition of such an excitable media, especially the brain, for example, and that has a few consequences because independent on the molecular level, which is by sure important, you can make a few statements to say, for example, that these systems are depending, precisely on external conditions. They are dependent on so-called weak external forces. That means we cannot predict, according to the fact that we have a weak force, where the system will go to. A few things which basically also although that has nothing to do with each other, a few things which have a certain similarity to some quantum physics aspects. We have uncertainty, and we have depending results or later results on small stimuli. It's quantum mechanics that would be the measurement process. So summing up these statements, I think first, when we accept that biological systems are these excitable systems, that they are following the laws of non-equilibrium thermodynamics, that there are a few things given, like, for example, systems far from equilibrium, open systems, these things which belong to the definitions, then we have a few problems, because then we cannot necessarily predict a few things. I had a very interesting discussion in this field, for example, years ago, with a very good neurologist, Wolfgang. I don't know whether he's still in the field. So Wolfgang made a statement which led to a very, hard discussion. He said sooner or later humans will be able to simulate a complete human brain. And this went to a point when a lot of people, including me, said, okay, maybe you violate a few very basic things. I even remember a discussion following that with Connie in how far even Gödel's statements would be involved in this point. So meaning, I know we have this good knowledge on molecular levels, we know a lot about molecules and what they do, We can go possibly 1 or 2 or even 4 steps further. We can look at the membrane, try to understand membranes on the molecule level and so on. But I'm absolutely sure at the moment at least, we are talking about the physics we know presently, that there might be a big problem to really go from the molecule finally to the brain or to the whole biological organism. I don't believe that, sorry.
Thank you very much. Thank you for your...
There is a big difference between you and me. There is a big difference between your position and mine, because I believe in it. And I think not only I believe in it, but I think it's necessary to do it in the present time. And I think to go along with you is a very difficult enterprise, that's for sure. But to even go beyond the water molecule or the membrane protein or the lipids, we have to make a multi-level kind of modeling, which not only take care about the protein, but use the proteins, use the knowledge we have at different areas. And I think this is perhaps what the challenge of the physicists could be in the future. I don't know if they want.
I mean, even if you go to a large scale, nobody wants to ignore molecular biology.
But everybody does. That's the fact.
No, just in microscopic series, the molecule features show up in a different manner, in my opinion. For example, what we measure in order to create a pulse in our models, when we measure compressibility. And of course, compressibility is integration over the properties of many molecules. Yeah, and the compressibility is, in some sense, a collective property of the molecules of which the systems are composed. But we don't need to know all the details of the system in order to measure compressibility. It's completely sufficient to need to know the compressibility.
You need to know what is relevant. That's it. And if you have proteins, the cooperativity of proteins is something which has been studied for 50 years now. And that you cannot ignore if you want to build into the Hodgkin and Huxley model the cooperative step that they very poorly represented in their computation, which is not even seriously being dealt with. Then you have to introduce the cooperativity of the protein in the membrane. But this is not done. Why? I don't know.
Okay, I would have to, I think Hodgkin-Huxley is anyhow, in my feelings, a little bit misinterpreted. But Hodgkin-Huxley is just a set of equations, and when you ignore the mechanisms behind, it's a very general description.
But many physiologists believe that this is a mechanism.
Yeah,
I agree with the critics which have been made. It is not a mechanism, it is a phenomenological explanation, and the mechanism is molecular.
Okay, but on the other hand, for example, the equations can be used in a modified version, for example, to describe waves in the brain, to describe waves in chemical systems. Basically, the equations is the same. And I even remember that in the early 60s, equations were used by people from plasma physics to describe oscillations in plasmas, which are more or less identical to Hodgkin-Huxley. So the point or the problem for me comes in when we have two sides of view. The one thing is we say we have really molecules which have decisive functions. On the other hand, we say we have membranes which as cooperative systems do something. I think both statements are not, as absolute statements, are not correct. We need both views.
I'm sorry I spent to do this kind of thing with f.l.t.r. So I don't want to say that we were correct, but at least we try to link together property of protein and the cooperativity of membrane made up of this protein. Now there are not many membranes like that, I agree with you, but some of them do exist, for instance in bacteria, where you have some receptive surface, chemosensitive surface, which is made up of about 1,000 molecules, and they all have together some phase transition properties that a single molecule don't have, or even when you put 2 or 3 molecules. Now, why this is rare? Because if you had such kind of monolayer of of proteins highly cooperative in a synapse, it would be stuck in a synapse for perhaps seconds, even more, hours or days. So that's the reason why this system is not used in the synapse. And if I may say, there are devices in the postsynaptic membrane such that they keep the receptor molecule distant from each other so that they don't interact. So that's my interpretation. But if you put them now in a bilayer or a lipid bilayer, they interact. And they give a lot of cooperativity with single channel on top of single channels, which is due to the fact that in the lipid bilayer they do interact, but in the membrane they don't.
You made a point that the receptor molecules want to stay apart from each other in order to be not cooperative. And that is something that I hear often from the biology side, to treat molecules as if they have intentions. They want to achieve something.
No.
But I believe that the molecule is fundamentally stupid. It doesn't know anything.
No, I think, sorry, this is a way of expressing myself, and I don't give any intention to a molecule. No, please don't do that with me.
Okay, so we agree that molecules are stupid.
Nobody does that. Yeah. I think there is an Aristotelian finalistic way of expressing things which is used by scientists just to express themselves, but which has absolutely no reality as far as their origin and their mechanism involved. I'm sorry to say that, but, and I'm sorry to have used this kind of expression.
Sorry, I don't want to accuse you of anything. But what molecules do in complex system is that they, as a system, strive towards the maximum entropy state. That's the only thing that they do. They want to become. They strive towards the most likely state.
They want to become you.
Yeah. Okay. Yes, you got me. That's true. It just happens to be that they in the end are the most likely state. But they do that with thermodynamic couplings. The different variables are coupled to each other, and we have the different thermodynamic functions, the intensive and the extensive variables. The proteins, for example, in a macroscopic systems would be included in terms of chemical potentials and the number of the respective molecules, and I think that's the right way to talk about that. And the system as a whole strives towards equilibrium. That's the only law that how would you say that enforces what happens in nature. I think except for increasing entropy, there's no other law. That's the only law.
Well, except that they are dissipative structure. And I had the opportunity to work with Prigogine myself for a while, and I think the way they are far from equilibrium, steady states, which according to him are absolutely fundamental in living organisms. We are all far from equilibrium. Everything in us is far from equilibrium. According to what he said, I know that Jacques Monod did not like this kind of reasoning, but myself I think it's acceptable and interesting way of thinking about thermodynamics of living organisms.
I would like to make two statements concerned to that. One is we can possibly bring it to the point and ask, is the classical ion channel really the ion channel within a protein, or is a protein modifying the environment, the lipid, insofar that it goes to the state what we need thermodynamically to get lipid fluctuations? Because, it's obviously that we have the proteins around, and so from this point of view, I think both points of view up to now are not really can be excluded. May not be really fair, but I think up to now no one has really precisely verified that this classical construct of an ion channel exists. On the other hand,
What
that
do you mean by that?
what I mean is when you have, for example, I have been
Ion channel have been studied at the atomic level now, and the translocation of ions through ion channel has been simulated by molecular dynamics. It's a whole discipline which is working on that. How can you say that?
yeah, no, okay, the simulations I accept, but the simulations,
How can you do best? The structure is known by X-ray crystallography, now by high-resolution electron microscopy, and the computations are made for trying to understand how the ions go through, which is not simple, I agree with you, but all these things are currently being done.
What do you think that is?
This is an old-fashioned electrophysiology. It's a voltage club.
This is just some channel event. This is a membrane
But
without...
fortunately, this was in the 70s, and now we are in 2020. This is 50 years ago, so 40 or 50 years ago, and I think science is progressing since these times, fortunately.
So what became better?
Nothing is better. This is one step which was historically very interesting and important, to have the capacity to measure single channel and record them in the membrane. And now we have to go much further than that. This doesn't give any explanation of how the ions go through.
Yeah. But this is a membrane without any proteins.
This
is a membrane without any proteins. It's voltage gated, it's outward rectified, it shows all features of a protein channel. But there's no proteins there.
That's exactly fits with what I said. It doesn't mean much. Thank you very much.
So, do we have the same opinion or an opposing opinion? I'm not sure anymore whether I'm in conflict with you or whether we have the same opinion.
About this, we have the same. In the sense that this single channel recording does not have protein with or without protein, with or without molecules giving channel properties to the membrane and so on and so forth. They are just a phenomenological observation, which are of interest for one thing to us, is the dynamics of the change of conductance. And here we have a view of how fast things are taking place and of how long the channel remains open. And we have to account for that if we really want to understand the regulation of ion channel in membrane, the physiological ion channels.
But for me that experiment just shows that in order to get a phenomenon like that, you don't require a single molecule fuel.
You don't need a molecule.
You don't need a single molecule to get
so
that.
please try to remove the ion channel.
There is no ion channel in there.
No, but precisely you are saying you don't need to have proteins to have the same phenomenon.
Yeah.
So it means that in this condition it's totally meaningless for biology. Because if you remove an ion channel, a real protein, from your genome, you are not going to stay here.
Okay,
this would of course be bad.
Not only if you make the knockout, as it was shown by Nobel, if you make the knockout of the ion channel, I think you kill yourself. So because the sodium channel is, well, there are several ion channels, it's not that simple, but whatever it is, it means that it's absolutely necessary. And more interestingly, I will talk to you more about it.
Okay.
In one word, some of these proteins, not this particular one, are present already in bacteria. And they are the same in our brain. So it means that there have been an enormous evolutionary pressure to keep them in living organisms, and since this simple mutation of these things would kill.
Let me get that in just one question about ion channels, because we almost agree, obviously. Just what is for you the most convincing? The most convincing experiment that the ion goes
That proves
through a
that
protein.
there's an ion going through a protein.
That the ion goes through a protein. and not
They
somewhere.
are, first of all, the purification of the protein, the reconstitution of the protein into lipid bilayer, and showing that the properties are preserved, for instance, for the sodium channel, the inhibition by tetrodotoxin. Now you have to go in vivo, make knockout, and see to what extent they contribute to the function in the nerve impulse. And I think these are a few things which for me are not only convincing, but show the absolute necessity of this protein for making an action potential.
May I, not a question, you see. I think the problem is not to negate the presence, for example, of sodium channels for example, in an axon. The question for me, and it seems to be the point of discussion here, the question for me is more the question of the fine structure of the event itself. Does the ions really pass through this pore within the channel, or does the protein just modify the lipid environment? And this is a little bit more difficult to say.
That's, I think, it's an interesting discussion. And where is the ion path through the protein? I think this is a really very well formulated question for the structural biologist who is interested in the function of the structure he is looking at. And that's exactly, we have a paper in preparation for that, so I do that, for receptor, which is an ion channel linked to an ion channel. And there is, I don't think anybody is questioning the way the channel is working and the ion is passing through the membrane. But the question is how the ion enter into the channel from the outside and how it gets out. This is, maybe you say, I can show you the data, and I can show you also the mutational work which has been done on that, which has been published. So it means that you have ways to approach not only the translocation of the ion through the channel, but where the ion enter into the channel from the outside environment and get out of the channel inside the cell.
There's a beautiful experiment by a former student of mine, Hanke. What he has done, he has reconstituted a potassium channel in an artificial membrane. And he used that membrane that everybody uses in the field. It's a PG-PC mixture, if I remember correctly, in a certain ratio. And he sent that lipid mixture to me to ask me, could you put that in the calorimeter? And then we put it in the calorimeter, and this lipid mixture showed a phase transition exactly at room temperature, where everybody measures. I'm not finished. And then
I am preparing the response.
Should I stop? The response to the question I didn't even ask. So he put a potassium channel in there.
Yes.
And the conductivity of the membrane followed exactly the melting profile of the membrane. The lifetime of the channel also followed exactly the shape of the melting profile, which is enforced by the fluctuation theories, because in transitions the fluctuations are slower. But he could at the same time show that the conductance is larger when you put more protein inside. That means it has something to do with the protein. More protein means more conductance. You could still block the event by the respective poison, and nevertheless, it was so that the complete physics that you saw there was a membrane physics. In my opinion, that is consistent with assuming that the protein acts as a catalyst for pore on the membrane, because you see the physics of the membrane, and if you add more catalyst to the system, you get larger conductances. That would not negate the role of a protein, but it would question the particular pathway for the ion.
Okay, but I have to. I understand the experiment. I think it's an interesting experiment in the sense that you tend to show that the lipids contribute to the property of the ion channel.
Yes.
And that I think everybody is going to agree with you. I don't think there is any disagreement. And the problem is, for the biochemist, or the molecular biologist we are, is to extract the channel protein from the membrane. Because if you want to reconstitute, you have first to extract it from the natural environment. And then you use what? Detergent. And detergents are known to alter the property of the ion channel. So you have to be very careful about the relationship between the lipids and the channel, and I agree with you, if you put more channels, you increase the overall conductance of the whole thing. That's for, it's not for me it's not a problem, it fits with the way we are looking at them, and I don't see any problem, but except that, as you pointed out, there might be different environments of a given ion channel in different parts of the brain or of the body, and the lipid environment is going to change from one very particular part. And even what I read recently in Nature, which is something astonishing, when you look at the lipid composition of the brain from the monkey to the chimp to humans, there is a dramatic change of the overall lipid composition of the brain. I don't know how it happens, because the number of genes is very small, but it happens. So it means that even when you work on some organism, the lipid composition may have a critical role. So I agree with that.
Okay, maybe this is a good time to switch to the next question. We have one-on-one.
It's still time until midnight.
Now that we all agree on ion channels, right?
I think we wanted to go back to complexity a little bit. And one typical statement about biology is that it's inherently very complex. It becomes perhaps even complexer with evolution, as you suggested. And we're interested in your points of view, is complexity a fact? Or is it also partly, where does it come from? Is it partly a consequence of starting with molecules?
Okay, basically I think complexity is a fact due on physical principles. When you define, for example, an excitable media and rising complexity just by definitions like open, far from equilibrium, a large number of small compounds interacting non-linearly and having feedback, then complexity is just a necessity. I think it's not even a construct, it's necessity. It's just that the scientific society had to accept that. The second point is most biological systems perfectly fulfill these preconditions. They are far from equilibrium, they have all these things, and as a consequence, might be also that they have the tendency to self-organization. I think when you go to evolution, for example, I know that this is not very much the thing what people like when they are discussing about evolution, but even there I think this question of self-organization and complexity, these are two parts of the same coin. This thing is necessary to understand why the biological system, the global biological system as we have it now, evolved and how it will evolve further. You see, when we would not have complexity and when we would not have this possibility, what I stated previously, that these systems in addition are critically depending on small external stimuli, that means anything which is living in a certain way, as we have even heard in the talks here, is connected to the environment. That was just what we had in this talk a few things ago. Then biology or biological systems interact with the environment and developing, following partially these stimuli. So complexity for me is what I said. It's just present. We cannot ignore it. It's necessity. It's there. It's a given. Physics tells us we have it, and so we have to live with it.
You want me to?
Yes,
Go ahead. I have to say.
I'm thinking about the answer to your
The word complexity reminds me of René Thom, who was a famous mathematician in France. I don't know if you heard about him. You did. And he said, well, people are using the word complexity when they don't understand what it is happening. So, I think it is an issue, and nevertheless, we have to understand the question and why you raised the question. And what I would like to say is that perhaps one can have an indication with the number of levels of organization. In the sense that, very interestingly, biological organisms has evolved from cells, first of all, and I may say that up to now, for reasons we may discuss, nobody has been able to synthesize and reconstitute a living cell. Not because of vitalist argument, but for other physical arguments, I think, which are interesting to analyze, but which are still totally unsolved. And whatever. You have the cellular level, then you have progressively in the course of evolution the addition of levels, which is multicellular, the organ differentiation, the brain, and so on. And interestingly, it looks as if, in the case of the nervous system, even if you have a very general plan for vertebrates, you can see that there is not only an increase of size, but an increase of the way the neurons are becoming organized on top of each other. And then you have to answer the question why this is happening and what can be, or I'm sorry not to use Aristotelian finalistic, but just to say to what kind of process does it correspond. And there was a question raised by this lady here mentioning Teilhard de Chardin, who was a Jesuit, which has more or less been excommunicated. He was a book which was called The Phénomène Humain, and which is I read recently, which is an interesting book, but I think where I cannot follow him is that he precisely describes the increase of complexity from alpha to omega, and he believes that there is an omega. So omega being some kind of highest level of organization of the human brain, which may be the lowest one after, in the next 50 years, I don't know how well we shall be on the earth in that time. But so I do not follow at all the issue of alpha and omega, but he has very clearly described this increased level of organization and the capacity for the living organism to invade ecological niches. So some of them are easily in contact with an environment which let them reproduce easily, and then others which are eating from them and so on and so forth on top of them. And human beings have, thanks to their nervous system, been not only to reach a situation where they can anticipate more or less the climate, anticipate the food income, anticipate the reproduction, and so on and so forth, but they are having such a brain for that they have completely invaded our planet. And if you look at the evolution of the human population in the next few years, it's absolutely explosive. So my view is that the increase of complexity gives a possibility to explore more and more kind of environments until the fact that at some stage there will be some limits, unfortunately.
I think your point on that complexity is often a sign of not understanding something. You know, it's like a kind of a cap word for not making any progress. It's quite that actually there was I mentioned this from Leibniz that he said that he would never trust anything is of the basis of a physical law if it's a very complex mathematical equation. So the more complex the equation gets, the more variations you have to fit into the same thing or different things. So he says it's a necessity that it's simple, that the mathematical equation should be simple in order to be a candidate, even from a math perspective, for a law. So I think this is something also that gives me hope that nerve pulse propagation might be described by much simpler equations than Hodgkin-Huxley. It actually describes actually more. The complexity comes from complexity does not come from the fifth parameter in this case, but from the laws and how the laws interact with each other. I mean, you get pressure, temperature, pH, and whatnot. If you start from sound, you do not get that list of 15 or 50 or 60 parameters of Hodgkin-Huxley. Yeah, but you must.
I think one has to distinguish between complexity and what is complicated.
Yeah.
But I think when many people, as you say, people say things are complex when they don't understand them, I think what they mean is it's complicated and we need more funding and more research time and more PhD students. But I think this kind of complexity often comes out of having the wrong angle from which you look at. I strongly believe that most fundamental laws are very simple. And the more simple, the better. And if some things appear complicated, then it's a sign for that one looks at the phenomenon from the wrong angle. When one uses the false premises. But on the other, when we come to complexity, in contrast to being complicated, I come from a department of biocomplexity. And these people actually mean something very structured when they talk about complexity. They mean self-organization, basically, in the Prigogine sense, far from equilibrium phenomena that self-organize. And that's what they call complex, which in my opinion might not be complex but simple.
I think there are two ways of responding or reacting to what you say. First is the actual organization of nature, the way it is organized, and the way we try to understand this organization. And I may agree with you that there is, as Descartes said, one way to understand and progress in the understanding of what we say is to find the simplest way to formulate it. And that's, I think, our role as scientists is to try to find simple solution to, let's say, complicated or difficult situation. And the next point, which is somewhat independent, is that you think that even if the situation is very complicated, you may find simple solution at the end. Yes. That's maybe I have no position about that. I think I am not disagreeing or agreeing with what you said, but maybe there are simple solutions because they are useful for the Darwinian selection to take place. And that selection has selected for innovation, and innovation might be simple. But this is just tentatively. I have no. I think it's a good question.
I think we really basically can agree. At least I have no problem with that. To say the rules that go to lead to complexity usually are very simple. Absolutely no problem. The question for me is additional to which degree of complexity can a system evolve. And I remember a discussion with Bohr. I don't know whether Bohr from Prague. He was working.
I thought it was the composer. There
were a lot of discussions, for example, especially concerned to this question, how far can the human brain evolve to which degree of complexity? And it was insofar interesting. Again, the rules are very simple. The way to complexity is, I think, quite well understood. But what is not that good understood is where does the system finally go? Is there a limit where the system is too complex and gets unstable again? And that's also a question, I think, basically related to what presently happens here in the whole biological system when you take it as a complete system. Is it finally going to a state of complexity where it's no longer stable? And then there is obviously a problem. And as a statement of Bohr, as far as I stated correctly, was that the human brain, for example, on his feeling, according to its complexity, would be already very close to this thing that a higher complexity would make so unstable that there would no longer be an evolutionary advantage of that. So finally, we're saying very brutally, we are at the end of the story.
Okay, I want to give the panelists, before we include the audience, the option to give a short final statement if they want to. And do you want to say some final words before we include the audience?
Before the beer.
Before the beer, yes. If you prefer discussions, if you prefer discussions for beer.
Let us continue to work all together.
Very good. Okay, well, I thank the panelists, and then you will have the chance to give if you have a couple of questions to the panelists. They can ask now. You can be seated. So you don't have to ask anything.
I have no question.
Anybody?
Two comments, mostly to Thomas. First of all, you talk about the great laws, everything being very simple. I once read a memoir by Francis Crick, who was a really great biophysicist. He said when he switched to space genetics, he had to switch his thinking completely and learn everything afresh, because he was looking at very small evolutionary changes in a glycosylation site or a small amino acid. So I think, great concepts are fine, but when you're dealing with evolution, you're looking at structures which are subject to evolution, you have to look at tiny changes. Another thing I think is also you're kind of about the single water molecule is really anti-reductionist ad absurdum. Because if we heard Dennis Noble talking about mutations in cardiac tissue, and we know that there are mutations which affect conduction in nerves. So if you're working on nerve conduction, you ignore that information, it seems to me not a smart thing to do.
You can give your response if you want to. Yeah. I mean, when I say things are simple, I don't mean they are simplistic or primitive or something like or trivial. But what sees in life is that permanently some repetitive patterns show up. For example, oscillations. Wherever you go, you find oscillations in the heart, in the beating heart cell, you find oscillations. And that seems to be, in my opinion, and the emergence of oscillations is what defines life. And everything that doesn't oscillate is not living. And when there is, but you have so many oscillations all over the body, and I think oscillation is fundamentally simple. It's a simple concept, even though the biochemistry behind it might be complex, but I think the phenomenon that shows up in the end is simple. And I find we have many of these simple, also in evolution. For example, I've seen beautiful presentations on the emergence of the spine. The spine has many bones that are very similar. That is a process, there's an oscillation behind that. You just repeat the same cycle again, and it forms many bones that look more or less similar. I think that is, in my opinion, something simple.
Whatever it is.
Just adding a short comment on what he said. This point of having more or less a problem of oscillations or repetitive structures, it's more or less a necessity of excitable media and these nonlinear systems. One of their outcomes is oscillation. So there is a very simple physical basis of this. You see, when you have a system which is, we typically say what is propagating waves, oscillations are more or less physically very well known. We have them in all physical systems which fulfill just these few basic rules.
And with oscillation, you lose a lot of information, don't you? Why? Because it's redundant and repetitive. And if you are trying to communicate through oscillation in your brain, you may have problems. I don't want to say that they do not exist.
Yeah. You mean I come back to the same argument again?
But this is one way of looking at it. The other way is that when you have neurons which communicate with oscillatory system, then you would like to have these convey some information one to the other. And if you have a completely redundant kind of scheme, then it's difficult. In fact, now some people are seeing that behind the oscillation there is some microstructure which is present there and which convey information. But I am careful about the oscillations.
Okay, but these oscillations, I think you can use them because you can modulate amplitude and frequency.
No, but I am not going to say that you are not going to use them. If they exist, they simply have been preserved by evolution and they are certainly playing a role. But I think to some extent the role of oscillation has been overestimated. In the brain, I am not talking about the morphological evolution.
All right. Okay. I think we are good. Oh, of course. How could I oversee you?
Is it a question or
You always it.
Is it a judgment? You are giving your opinion?
Well, of course I have opinion. But, let's start with something which exactly is on this type of discussion and is exactly making the point of anyone here in the panel and is even accepted by the public since the whole process of the problem has happened 200 years ago when Leonhard Euler brought up the acoustics, and the whole theme of acoustics of music was solved. And I put just music and you will see the complete parallel in all psychological details and differences which have been brought. Suppose Gange and Thomas listen and don't... No.
You have to reprimand it.
No. And this is now, I talk to every one of you, and I talk to you since you know all that. Music is in the air. I ended my thing by saying music is in the air, just like nerve excitations in the lipid bilayer. But we stay with music. The air is simple. You can exchange. The air is thermodynamic. It's the first derivative of entropy, which makes the sound wave propagate oscillations. It's the second derivative, which makes the Brownian motion, since now actually we know. So air is very simple, and you are stupid to say music is in the air. You cannot have a music without the instruments. You cannot have a music without compositions and musicians, and you are totally stupid to put into this very simple air or water or whatever you have, the Jupiter Symphony and so on. So now let's go through the state when we did not accept that music is in the air. Music is with the musicians. And now Thomas comes, or Wolfgang comes, and says, we can take the instruments away. Yeah? And then of course Jean-Pierre will say, you're stupid, you cannot. They are there always with the music. Well, no, we can do it with a loudspeaker from a CD. But maybe at that time we could not. And now we come to the point of genetic point mutation. So you make a point mutation. Make a point mutation in the flute, drill a little hole, and everything is different. Everything is different. And now you go to the musicians, they won't listen to the whole thing. And this is the combining point. The musicians and the composers, they compose with the brain and not with the laws of acoustics. They compose with the brain. They are creative, and this is the composing thing. Gange, I'm explaining you something. I'll explain you something. So the combining point is the following, and this combines all of you. I don't have the time now. You all do exactly the same thing once you take the point of view of composers. The bossa nova will agree with the march of the military and with the Beethoven symphony. Totally, it's the same brains which do it, but in a totally different. I have to look this way because they are the undisciplined. Now I make my conclusion and I make my claim, and tomorrow we will have more of that. We conceive our ideas. This is my pedagogic trick. We conceive our ideas with the brain. They have always been conceived. And the laws were always simple. And what we are living, I mean, if you go through the Copernican revolution, sitting not on our planet, if you go through the Einstein revolution, sitting not on our time-space, but on light, and we are on a revolution of the same scale. So we can expect that there may be still centuries for it, but in all cases, we have very good reasons to believe that Copernicus was rubbish as to observation in the sky. The Ptolemies, they had everything, he had nothing, experimentally. And Einstein was rubbish still today. Yeah, now it's your turn. You reply. Let me finish my point.
Yeah, you get your chance to talk as well. It's not just, you know, just a second. I think we got the point. The music is very important. I think we see the parallels between that, and we can continue that. But I'm saying all of us deserve some food and some drinks in which we can continue and think and talk about this, and maybe even play us some music tonight, which you were lacking yesterday. For this, I'm going to reprimand you now. Okay, with no further ado, I welcome you to join us in the basement with drinks and food from right about now on. Okay, so thank you very much and thank you.